there in existence and used quite commonly. So next slide. So for the particular case of stirred tank reactors and cultivated meat, so there's a number of different starter cell types under consideration for cultivated meat, but a large grouping of them are what's called adherent, which means they must adhere to a surface to grow, to proliferate, and all that kind of good stuff. And so one way this is sort of achieved in these stirred tank bioreactors are using these small little bead-like objects called microcarriers. And there's a schematic of this process on this slide, and you can see all the cells that are adhering to a small little sphere. But the problem with this is that it renders the attached cells more sensitive to potentially damaging shear forces and turbulent flow. And then at larger scales, you're going to have to mix the system more vigorously to achieve the kind of homogeneity that you need. But that, in theory, can create more damaging shear forces. And so having these two kind of limitations in mind and knowing there's a trade-off between stir speed and shear forces, it kind of begs the question, well, how do you scale such a process? And one way to do that is to build a model, which is what the last part of this slide is trying to emphasize. So next slide, please. And so this slide here with all the little spaghetti wirings and whatnot it's not actually a microcarrier system, it's a microbial system. But it illustrates the point that, bioreactors, which have a lot of control, are quite complicated. So in this case, there's a whole host of different toggles or elements of the system which you can toggle to optimize the output. But if you wanted to create a model to encapsulate all these different aspects, that's going to be complex, and switching one element of the system you're going to have to understand how that impacts another. And so to create this sort of base model, it seems more important to start with something much simpler. So next slide, please. And that's precisely what we did for this proof of concept work. And so I had identified this older paper from 1987 by an MIT researcher, and the very simple bioreactor design that we considered is here on the left. And it's just simply a spinner flask with a little stir bar in it. And there was obviously cell culture media in there, and then the cells bound to microcarriers, and it was kept in an incubator. But what this paper did, kind of in isolation, was look at stir speed and then cell growth. And this seemed to be a great target to unite these two modeling platforms that we were interested in. Computational fluid dynamics, which could very accurately model stir speed and all the fluid flow components of the system, and then agent-based modeling, which could understand the cell growth and all the sort of forces that the individual cells experience. And so we took all the information from this paper from the 1980s, and we used that to build this foundational model, which was much simpler. And that's the end of my bit.
So hi, I'm Jiajia Chen. I will introduce you, as kind of Greg mentioned, our first step of the modeling process is to develop a computational fluid dynamics model to simulate the flow field in these simple bioreactors. Next slide, please. So let's first look at what's the computational fluid dynamics. So computational fluid dynamics is also known as a CFD, which is a very kind of well-developed branch of the integrated disciplines of fluid dynamics, mathematics, and computer science. So CFD is kind of a general approach that uses numerical analysis and data analysis to analyze and solve the fluid flow in different types of bioreactors. And the CFD has already very well known to use in the different applications, such as to design the airplanes, the automobiles, in terms of to understand how the different equipment or different transport system will behave in the air flows. And similarly, actually, the CFD is also used widely in the bioreactors simulations to understand how the flow behavior in the bioreactors. Next slide, please. Then here, actually, just as Greg mentioned, our CFD model tried to simulate a very simple case from the published paper, Graham 1987 paper. The first step of our CFD model, which is to use their reported geometry, and we create the geometry in our software, and the software we are using here is a finite elements-based commercial software console. And also there are other types of software available, commercial software available, such as Fluent, which is using the finite difference approach. But here we are using console, and different methods, they will solve the CFD problem similarly. So after we develop the geometry, then we discretize the whole geometry to be millions of elements. This elements is kind of like a small parts of the geometry. And on each element, we may have multiple nodes. So we can calculate or we can simulate the flow behavior, like the velocity, the shear stress, etc., on each one of these millions of points on the nodes. And we are using this package to solving the different type of governing equations on each one of these nodes. And these governing equations generally for the fluid flow, which is Navier-Stokes equations, as shown on the right side of these governing equations. Next slide, please. So for all these models, for the CFD models, we actually can, using the different solvers to simulate the different scenarios. For example, on the left side, we can simulate the steady state flow, which means if we mix the bioreactors for enough long time, how the flow field will behave inside of the bioreactor. So the velocity may change periodically at the different locations, but generally this change will not change with time. So that's called the steady state flow. We also can simulate the transient flow, like at a different location and a different time in the bioreactor, we can predict how the flow behavior behave inside of the bioreactor, like what's the velocity, what's the shear stress, etc., all this kind of stuff. So next slide, please. So in our model, next slides, please.
Okay, I did advance the slide. You should be seeing effective impeller speed velocity.
Okay.
Is that what you
see?
see?
I see it now.
Okay.
So in our model here, for this simple model, what we did is actually using the steady state model, because the advantage is that it can predict the results quickly, because it's a proof of concept model, and we use a steady state condition to simulate the fluid field. And we evaluate the different velocity, the different impeller speeds on the flow behavior. So here shows two different impeller speeds, 60 RPM and 240 RPM. We can see that the velocity profile looks very similar to each other, but if you see closely, actually, if you see the legends, the color legend of these two files, they have the quite different the velocity, the magnitude of the velocity. Okay, that's outputs from the CFD model. In addition to this velocity, we also can simulate like the shear stress, the dissipation energy, etc. All the different type of parameters can be predicted by this CFD model. And for the next step, Yaro will explain how the ABM model will use the CFD model results in the biological process modeling.
Hi all, here's Jaro from the Netherlands. I'm affiliated with the University of Amsterdam and the Netherlands eScience Center. And first of all, so I'm going to talk about agent-based modeling part of this proof of concept study. First of all, I want to mention that most of the credit goes to Boris Egler, who did most of the work on this, but he sadly couldn't make it to the meeting. So next slide, please. So a bit about agent-based modeling, in short, ABM. So agent-based models, in agent-based models, one simulates the actions and interactions of autonomous agents to assess their effects on the system as a whole. And specifically, emergent effects are very interesting. So, for example, cell sorting in the image you see below, which simulates tumor growth, for example, with the Biocillion framework. But it's also used in other biological systems. For example, a study from the University of Amsterdam, where they modeled the gastrulation of a sea anemone during its early development. So the point is that you simulate agents which are autonomous systems that can interact with their environment and other agents. But they are autonomous themselves. And it can, for example, also be applied in many different fields, like you see the image of the evacuation planning, where they modeled a crowd of people wanting to leave the room through one exit. And the output is how many people actually left the room and how many were injured. And it seemed very unlikely, but apparently, if you put a column, which you see in the bottom image, just in front of the door, more people will survive and less will get hurt in the end. So this, in the end, was used to optimize emergency exits by putting a column just in front of it. So next slide, please. So we use agent-based modeling to model the muscle cells and the microcarriers. So we have two types of agents, cells and microcarriers. And cells like to adhere to microcarriers and to each other. So all of these agents have several characteristics and can be in a certain state. And they experience forces by each other or their environment. For example, deformation force by each other. If they get too close, they get squished together and they repel each other. Or by the fluid flow, they experience a drag force. So we took the fluid flow field from Jaja's CFD simulation, and we put all of these little microcarriers with cells on them in this fluid flow, and let this run for some time. And you can see the result of that in the video. So next slide, please. So here we have the results from the simulations we run. So on the left, we have these two different impeller speeds that Jaja showed. And you see that with a quite slow impeller speed, we have faster cell growth, of course, because there's less stress on the cells. And with a faster speed, we have more stress, so also slower growth, because these cells get stressed and proliferate slower. But maybe a more interesting result was when we started to tune the stress parameters. So these cells, they experience force by either the velocity field or each other. And this results in a stress in the cell, and we made it such that these cells, that their proliferation rate depend on the stress they experience. So we have a stress parameter, and when they experience these forces, the stress parameters will define the proliferation rate of these cells. And you see, if we tune up the proliferation rate, the only thing that matters is actually the fluid flow. And you see almost a straight line. If we make it a bit lower, this parameter, you see at some point that there's the slope changes at some point. This is an interesting result, which happens when the microcarriers fill up with proliferated cells, and these cells start to experience each other's presence, and they start to push into each other, adding an additional stress to the fluid flow. Next slide, please. So this approach shows that that our approach, well, sort of works. Qualitatively, we see the same results as we had in the Krugen paper that Greg showed. But there's, of course, much to improve, because this is a really simple model, and we can add lots of different additional states or interesting effects in these agents, these cells. For example, we still need to add gravity to this equation, because that will pull the microcarriers down. Collisions between microcarriers could result in cell transfer from one to one bead. And there's, of course, much biology that we didn't model here, but which could all be implemented in the cell, so that the shear stress will influence the gene regulation network or accretion or nutrient consumption in these cells. But thing to mention is that if you add more complex models or add more stuff to the model, of course, this comes in with a cost on the computational side. So we need to decide which ones to add and which ones not to add. And that's the future perspective for this this part. So then I'll give the stick to Simon again.
Sure. So so you've seen the description of the setup, the scenario, how computational fluid dynamics results in these velocity fields, and agent-based modeling slurps up that information to predict the outcomes for the individual cells. So I wanted to talk about what the software pipeline looks like and where you can get the source code, show you kind of how the whole thing goes together to produce a like a demo, an interactive demo you can try, and quickly go over some possible application areas. And then I'll talk very briefly about the GFI grant. So next slide, please. So to begin with, we have this bioreactor geometry, which is generated by some domain expert. Well, creates a beaker or whatever that bioreactor might be, a tube, and feeds that into COMSOL. Next slide. Then COMSOL generates that fluid velocity field that actually has to be turned into a structure that the agents can find themselves in. They need to locate themselves in some sort of structure to figure out what the velocity is at their particular point, because as Jaja mentioned, there's a mesh that COMSOL uses. So there's software that's available for that. That feeds into Biocellian, which is an agent-based simulation modeling and simulation system, attuned to the needs of biology. That also has to take into account cell-to-cell biomechanics. That information has to come out of papers and resources, domain experts. So that's another input to the pipeline. And that microcarrier, ABM microcarrier model can be found on GitHub as well. Next slide. Okay, so the idea is that we're building out this pipeline. It's the software stuff. It doesn't know anything about biology per se, or about bioreactors. That other stuff needs to be fed into it, and that's where there can be proprietary information. So that's the pre-competitive technology is building out along the bottom, and then the technology that's specific to the problems, specific to companies, and so on could come in. Next slide. So finally, the Biocellian simulation spits out the cell microcarrier counts, their positions, their stress numbers. And again, that has to be pre-processed before it can be used for visualization. So there's some other software that's available on GitHub for that. And then there's an interactive visualization tool that we built, also available for download on GitHub. And we'll do a quick demo of that. Next slide. Okay, so you can go on our website, you'll find this. You can run it in your browser. There's a browser version. You can zoom in on this, on Kruen's stir tank reactor, and you can see the microcarriers floating around with cells attached. There's a number of different things you can do, like you can type F to focus on a particular microcarrier, like that. You can jump from microcarrier to microcarrier, zoom in on it, and see the stresses as they change. Red means higher stress, blue means lower stress. as it floats through. So that can just give you some idea of what's happening as another means of feedback besides just graphs. Not need to run it again. Next slide. So possible application areas. You could think about maybe in the future looking at how to look at different tank sizes and shapes and rotors and different kinds of bioreactors, looking at the stresses of the cells, how mixing occurs. We're not doing any mixing here in this example. We don't even have any media in this simple scenario. People have talked about looking at different types of microcarriers. Some of them, maybe changing the microcarrier shape would reduce the stress on cells. Some people are looking at even microcarriers that are soft or edible. The bioprocesses is another thing people have raised as a possible application area. There's a whole process you go through. Some people try changing rotor speed, some add additional microcarriers, and so on. So looking for an optimal schedule. That can also be modeled and simulated. And of course, the scale-up issue of how large can you make a given bioreactor design, and those bioprocesses, how do they need to change as you scale up without having to run so many laboratory experiments. Next slide. So that was the proof of concept. That was this past 6 months of work for our little team, sponsored by Merck. And going forward, we have, in addition to Merck's continued sponsorship, a grant from the Good Food Institute to look at novel designs, not necessarily stirred tanks, but we propose to look at rocking bioreactors, hollow fiber bioreactors, continuous surface bioreactors, and see how our pipeline needs to change for those. See how well we can characterize what determines their performance and efficiency. Do our best to design some multi-scale models similar to this one, and see what we can do to analyze the designs through our models. So next slide. I think that's it then. Thanks.
So now I'm going to stop share, and let's just take a few minutes here for any questions that might have come